Hi guys, this is Miss Nehad, the biology teacher from Bio with Nehad. Our topic today is about genetic code. Let's get started. Genetic code actually is matching the sequence here, the nucleotide sequence of the mRNA that it's already transcribed from the DNA in the nucleus, if you remember, just matching it with the corresponding amino acid to give by the end of the process of protein synthesis, the one we already talked about in the previous video, to give the protein at the end. Just to remind you with what we have been talking about in the last video, we were talking about the protein synthesis. Protein synthesis, it has two steps. The first one is transcription, the second one is translation. Basically, the transcription, the first one is about making a copy of mRNA from a DNA template. So this is the DNA template. A complementary copy will be transcribed here. This is the mRNA. Then the second step is translation. The translation is changing, converting that mRNA into amino acids. How? By just decoding each three nucleotides or nitrogen bases into one amino acid. The amino acids will be connected to each other or bonded to each other by a peptide bond. By the end, we'll be having a long chain of protein. So here, the mRNA will leave the nucleus and it will go to the ribosome, which is actually the location of the protein synthesis. Okay, and then the this subunit, the large subunit and the small subunit will be attached together, the mRNA in the middle, and that ribosome will be reading this sequence, the nucleotide sequence, and converting it into um, amino acids. So just think about it. It's like the question time. If this tRNA came and brought that amino acid, how can this tRNA just recognizes that sequence? Okay, how can it know that this codon corresponds to that amino acid that it carries? Actually, this is because of the anticodons. So the codons, the three nucleotides in the mRNA, they have three complementary nucleotides carried by the tRNA. So the tRNA is so specific. The one carries this anticodon, carries this particular amino acid. So whenever the codon comes to the A site, it will will attract the tRNA that has this anticodon. So here we have CGG. So the anticodon will be GCC, and the GCC always carries this amino acid. Okay, arginine. Clear? So guys, if you think the anticodon is actually the DNA that we started with, because the mRNA was um, the complementary copy of the DNA. Then by the end, the mRNA again met the tRNA. Okay, so the codon of the mRNA met the TNA, the tRNA, the anticodon. So the anticodon is actually complementary to the mRNA. So the anticodon is actually the RNA version of the DNA, the original DNA at the beginning in the nucleus. Actually, this chart shows us how to get from a codon the corresponding amino acid. Okay, so I have here the G GCU. Okay, so the first letter is G. So I'm here. I just go to the whole thing. So I have all of this. Then the second one is C. This is the second letter. So here was the first letter. This is the second letter. Let's change it into green. So this is the second letter. The second letter is C. So I'll just highlight that one. So which one is the meeting point? Now I restricted it into that one. Why? Because it's the meeting point of 2 and 1. Okay? Then number 3. The third letter is U. I'll go to the third letter in our area and it's U, so it's T here. Alanine. Let's try another one. So here I have AGC. Only 3, okay? So just remember that the codons, the mRNA codons, it's the only 3. So here ACG, the first letter is A. So here, this line. Then the second one is C. Second one is C, so that column. So it's restricted already to that one. And the third one again will confirm, which is G. Where's G? G is here. Here. So it's threonine. Okay? Threonine. Threonine is one of the amino acids. Okay? The point is the mRNA has a start codon, which is AUG. Where is the methionine? So this one. So the methionine is AUG and it's always the first one, the first codon of any mRNA. So the first amino acid of all the long protein chains, it's methionine. And we have 3 stop codons, TAA, TAG, or TGA. So you can just because it's mRNA, so it's UAA, UAG, UGA. Okay, so one of them, when the translation reaches one of those 3, it will stop translating that mRNA. So this is the stop codon, the sign that you have a stop. You have to stop here. So here the mRNA started with AUG, which is the A. So this one, the whole one, then U is that one, and G is that one. So it's the methionine. And we have this codon wheel. It's the same idea of the codon chart. We can get from any codon, we can get any amino acid corresponds to that codon. So we'll just start one example, AUG. You know that it's the start codon, okay? And we know which amino acid, but now I just teach you how to get it from the chart. The first, here the first, I'll just make it highlighter. So yellow highlighter. Here Okay, here this internal circle in the center, this is the first one, the first letter here, A. So I have to, this one, I need that part. Then the second is that wheel. So the second wheel is the second letter. So the second letter is U. So here after A, U. So I am here. Actually, maybe it's easier than the chart. Then G. The G is here. So A in the first circle, U in the second circle, but the U in front of the A. Then G. Say so. Here we have the AUG. AUG. So the first circle is the first letter. This is the first circle, the first letter. I'll just get the blue. So this is the first circle. It means the first letter. And the second circle here is the second letter. And the third circle, oh, here is the third letter. And if it's A, the first letter, I have to be only here in that, zone. Then I went to U, so the one with A, okay? Then to G, the one in front of U. So I have just to go from, small to smaller till I reach the last one. Let's do it again, but on the wheel. So G, I am here. So I have to go to G or A or C or U, only those. The second one is C, so I have to go to only C. The third one, and I have to go only in front of the C. Okay, so the only one here is U, so T here. So it's actually all of them are alanine. Okay, so the first one is alanine. Another color to go to the next one, ACG. So the first letter, the central circle, A. In front of A we have the second letter here, it's C, so here we'll go to C. Then the third letter is G, go to G. Okay, so this is the path that we have to follow. Number codon 3, it's G, C, G, A, G. So we're here again, G, second one is A, G. Okay, this is the one. We need another color. Okay, C, U, U. So here C, I have to go to that zone only. So the second letter is here, U, and then that zone, only one of those. Okay, one of those. So the third one is U, C, U, U. So leucine here. Okay, pause the video and these are the answers. Just practice one time on the chart and practice one time on the wheel, then go to that. These are the answers. Thank you for watching. Just wait for the next video. Subscribe and like. Thank you.